our regular Wednesday seminar series. So today's speakers is Professor Kubongyong in the IBS. Let me just briefly introduce his career. He got his a bachelor, master, and then PhD in postdoc in the Department of Molecular Department of Life Science. 분자생명과학과. He graduated 분자생명과학과. All right, and then after the 2 years postdoc in the same university and Seoul National University, he moved to the Hublet Research Institute. The institute is famous for the organoid, lead by the Hans Kleber. I guess you probably heard about his name because he's most important and then famous research scientist in doing the organoid research. And after a few years postdoc in the same institute, he moved to he got his position in the Cambridge Stem Cell Research Institute. Sorry, I only have the Korean CV, so I'm not sure whether the name is correct. And currently he's working in Austria, IMBM Research Institute as a group leader. Also, he's the vice president of the IBS, the 기초과학연구원 in Korea. So currently he present in Korea, and as you probably can guess, he is working on the stem cell and organoid field, and he published many important and interesting papers. So his original research, the topic he's going to present is about the gastric cancer, but the same topic has been presented by Dr. Han last week. So he suddenly changed his topic. But I guess his new topic would be more interesting and then more impressive to the student. So please, Professor Ku, the mic is yours.
Okay, thank you very much for the very nice introduction. Let me share my screen first. Okay, I guess everybody can see it now. So firstly, I should have been there in person to meet you all, but there was a family issue that I couldn't avoid, so I had to stay home to look after my big daughter. So I'm very sorry for that, and yeah, I mean, sometimes things are really not easy to get away from. So, and secondly, initially I was thinking about presenting a story about stomach stem cells and its plasticity, but I just heard from Professor Kyung-kyu Kim that Seung-min Han has presented this work last week, which is very good. So I'm going to present another story that I have, which can also be very interesting. So let me start. Okay, I will make this one smaller. Right. So instead of the stomach stem cell story, I'm going to present you about advanced genetics. And this is about how we do genetics in an advanced way in order to help the understanding of human health and human biology. So there are mainly two topics. First one is about mosaic analysis tools. And secondly, I'm going to also show the short conditional introns, which can also be used for generating conditional knockout mice using CRISPR-Cas technology. These two work have been led by two brilliant scientists. Firstly, Mingyu Yeom. He is currently a assistant professor at KAIST, so he became a neighbor of mine, and yeah, I mean, he's a great scientist. And the other part was helped by Sam. And Sam is a Taiwanese student that I have at the Vienna Institute, where I'm also affiliated. So I especially thank both of them because they have done a really hard work to make it possible. Okay, so before we let me find a way to hide this panel. Yeah, this is now better, I think. Before I get into the advanced genetics for my mouse system, I will briefly mention about the organoid system because many people know myself as an organoid biologist. The reason that I was interested in organoid was because it is a very nice model system. So in biology, people have been using invertebrate models such as C. elegans and fly, and the reason behind is that these are very simple organisms, and genetics is also very easy. However, they obviously have a limit because these are invertebrate, so there are many similarities, but there are also many differences. Because of that, people started using zebrafish and has been also using actively the mouse system in order to understand the vertebrate biology, which is more close to the human biology. And for human biology, nowadays we have an organoid system and many other ones. But when I was a PhD student, we only had cancer cell lines, which have many mutations, etc. So this was a main hurdle in biology when we want to understand human system. However, now we have an organoid system, and I can tell you that organoid is something that is sitting in between this mouse system and the human cancer cell lines, because it's an ex vivo system where you see an in vivo phenotype, and they are working almost like a cell line. You can also preserve them, which means that when you want to go for vacation, the experiments can wait for you. And it is also very easy to treat with ligand and inhibitors. And you can isolate mouse or human organoid. So if you are working with a human organoid, basically you can perform a human experiment in a dish. However, about 12, 13 years ago, when I started working with organoid, we didn't have a means to do genetics with the organoid system because they are growing in 3D and the culture is much more delicate. So it was hard for us to know how we can do the gene transfection, viral infection, etc. So because of that, from that time on, my main focus was developing genetic tools for organoid system. And I'm going to briefly tell you about what we've been doing. And firstly, we have established viral gene overexpression and knockdown method, which has led then to bacterial artificial chromosome transgenic organoid, which means that we figured out how to introduce 100 to 200 kb size DNA plasmid into organoid system, which means that we have established very robust transfection method. And because of that, we were able to apply CRISPR-Cas system almost immediately after the first introduction of CRISPR-Cas9 system. And with that, we were able to perform genetic knockouts in organoid system, as well as even gene knock-ins and gene corrections in human patient organoid. So in this paper, we have shown that cystic fibrosis patient organoid can be corrected by CRISPR-Cas using the intestinal human colon organoid system. So although we couldn't cure the human patient, but we were able to cure the genetic disease in human colon organoids. And after that, I became an independent researcher in Cambridge, and in the field, Toshiro Sato has introduced this NEPA-21 electroporator as a very nice device for high transfection efficiency in organoid. And using that system, my lab has introduced this piggyback system as an alternative for retroviral or lentiviral gene overexpression. And it also has a clear advantage over a viral system because we can carry not just about 5 kb of cDNA. Piggyback can carry even up to several hundred kilobase DNA plasmid into the genome of an organoid, because this works as a transposon. And we have also developed conditional knockout method using CRISPR flip method, which was published in 2017. And now people can even perform CRISPR-Cas screening in organoid, which means that we can also do forward genetic screening in the organoid system directly. Having said that, what I can mention now is that what you can perform with a human iPSCs can be done also in human organoid. So with all these methods up until now, what I feel now is that my role as a genetist and that person who developed technology in organoid is nearly done, because I cannot really think about anything really new for the organoid system. So from 2017 on, after I relocated my lab from Cambridge, UK to Vienna in Austria, I started thinking about shifting my topic from organoid to mouse genetics again, because I was initially trained as a mouse geneticist during my PhD time, and I've been always loving with the mouse system because it's such a nice system. However, we should know about the reason why people started working with a human organoid. The reason is that mice is not human, and because of that, we can now recapitulate everything in the mouse for understanding the human biology. However, organoid has an obvious disadvantage, which is that we are only culturing the epithelia of an organoid or some mesenchymal cells and immune cells of a certain tissue. And because of that, you basically lack the systemic effect. So you can understand the microphysiology of the organ of your interest, but you don't understand the entire biology of the system. So I was thinking that it's also time now to improve the mouse genetic system in order to mimic the human biology in a better way. So firstly, I thought about limitations, because if you want to tackle on some important questions in biology, I think it is very important that you first think about the limitation. So in the mouse, one very obvious limitation is we are performing genetics in the entire body or in the entire tissue. But for example, there are many hereditable genetic disease in human, but this is not all the disease that we have in human. Most of the disease that humans suffer are late onset disease in the adulthood. So for example, cancer starts locally from a single cell. And when we model cancer in mouse, we basically knock out tumor suppressor or activate oncogenes in the entire body of the mouse or the entire tissue. So because of the reason, and to model this in a better way, we started thinking about developing mosaic genetics too. And secondly, yeah, aging is not happening in a uniform way. It's also a very stochastic event, and that would also require something like a mosaic genetic system. And secondly, mice are not humans, so we can still use human pluripotent stem cells and organoids, but they lack the physiology that we want to have. So at some point, I think a primate model system will be very popular, almost like a mouse system in the near future. However, in the primate system, we don't know how to perform conditional knockouts. So for that, we have developed a short conditional intron method, which is currently working in the mouse, but which can be translated to primate for the conditional knockout in primate model system. And the third point is also a very important point. So in genetics in the mouse, we always perform only the knockouts. We don't try to rescue the gene. And because of that, many students and also many researchers, even in big pharmas, we think in a way that just by simply removing the cause, it can be a cure. But this is totally wrong because, as you know, in chemistry, I mean, this is the Nobel Prize of this year, that there is an irreversible reaction, which is like a click chemistry. And biology is basically based on chemistry. So in biology, we of course have irreversible reactions, which means that, for example, if you have a dementia, initially, this can be cured by removing the cause when it is starting. But once you have lost all your neurons in your brain, then you cannot cure it simply by removing the cause. You have to transplant neurons instead of that. So to study this kind of reversibility, I think it is very important that we also develop something like a reversible genetics tools. So this is an ongoing work. So I don't have a data today, but I wanted to share this important topic with you as well. So there are three main topics that I want to study today. Firstly, mosaic overexpression system, and then I will introduce short conditional intron system. And I'm going to show you how the first and the second system can be combined to be able to perform mosaic genetic knockout in the mouse. So before we get into the mosaic overexpression system, let me introduce the cancer development and the current focus of cancer biology. So cancer starts from a single cell because we know that they are clonal disease. However, the study of the cancer usually starts from the formation and invasion and metastasis. And we all know that even though we have been studying cancer for like 30 years or 40 years, there are many miracle drugs, I admit, but cancers are still very aggressive. And we know that their evolution power is too strong. So very often we see recurrences. And because of that, I think it is very important for us to understand the precancerous transformation biology of the cancer. However, this part has not been studied very well because the cells that are getting this mutation firstly in human tissue cannot be really easily identified because they just look as similar as the other ones in the tissue, which means that these mutant cells are histologically almost identical, so you cannot find them. And if you don't see them, it's nearly impossible for you to study them. And secondly, genetic modification has been done in a clonal manner, has been done in an entire tissue or even in an entire body. So we have to perform clonal genetics. So to be able to overcome these 2 points, we have developed a method to fluorescently label wild-type cells and mutant cells simultaneously. And this also helps us to do genetics at a single cell level. And if we have such a tool, now we also have a single cell analysis tool such as single cell RNA-seq and single cell multiomics. And because of that, once you have mutant cells, wild-type cells, immune cells, and fibroblasts, you can isolate this small biopsy. And after dissociation, you can perform, for example, single-cell RNA-seq to know the transcriptome information of individual cells within this small lesion. And by doing so, we can understand how this mutant cell is growing and how they interact with other cell types. So this sounds very much like a dream. And to realize that, we came up with this idea. So there was already a very nice lineage tracing cassette called Rosa 26 Confetti Allele. The reason that is called as a confetti is because it has 4 different colors within this lineage tracing cassette. So it has a stopper here. So initially there is no fluorescence protein expression, but once the Cree is activated, they will recombine this Loxp site in a random fashion. So individual cell will be labeled by either of one of the 4 colors. So you can basically make the whole intestine like you see this picture, which is very beautiful. This has been used by stem cell biologists who studies, for example, other stem cells in various tissues. And we were thinking to utilize this confetti allele for mosaic genetics system. So, and the way we have achieved that was by introducing this cDNA in the downstream of RAP. And by doing so, now what you have is red to cDNA system. And here you have the same stopper. So initially there is no expression, but once this stopper is removed by Cree, and then further recombination to choose one of the 4 colors, then among the 4 colored cell types, all the other cells are wild type cells, and the red cells are the one expressing cDNAs together. And by introducing, for example, oncogenes into this red clone, you can perform now fluorescence labeling, red clone expression of oncogene, and this is compatible with the single-cell RNA-seq as well. So you can visualize the cells to perform histological analysis, and at the same time, you can perform single-cell RNA-seq to know the transcriptome information. So we initially made 3 different mouse systems. The first one is Notch introducer cellular knock-ins, KRAS oncogene, and PI3 kinase oncogene. So here I have an example of KRAS, G12D. So at day 2, after the recombination, you see very nice salt and pepper pattern of yellow, red, and cyan cells. So they represent, very with a very nice ratio. And here, the labeling efficiency of yellow and red are very comparable. However, if you wait about 2 weeks, you can easily see this clear domination of the red clone because of the expression of KRAS in these red cells. What is striking to me is that the crypt-villus architecture of the tissue is still pretty much well kept, but the KRAS clone is fully dominating in this normal-looking tissue, which means that even though the tissue is normal-looking, and there is no tumor formation, the oncogenic clone can spread. So we made some validation. So where the Notch, KRAS, and PI3 kinase oncogenes are expressed, we have a specific downstream activation, like Notch ICD formation, phospho-ERK, and phospho-AKT activation. So this was a clear confirmation of the system. And the other beauty of the system is that the confetti system had a certain ratio of the recombination of yellow, red, and cyan, and that was also pretty much well kept in these other systems that we developed based on the confetti system. So now what you can have is a chance to compare each of the mutants to the confetti as a control. So we first looked into the clonal behavior of the wild-type clone and the mutant clone by looking at the crypt base. So this is the intestine. You have a villus, and in between you have a crypt toward the body side. And at the tip of the crypt here, we have a stem cells, and they are competing each other for the niche space. So over time, if you label one of the cells genetically, you can see their spread, the spread of their progeny throughout the circle, what I mean is there is a clonal growth. So if you label this single stem cell, it will generate 2 daughters, 3 daughters, etc. over time to make the entire crypt with its own progeny. And this is done by stem cell competition within the crypt base. And there is a beautiful theory behind it, and it's about 5-6 papers. So if you like to know more about this, I would recommend to read those. But if you think in a very simple way, you can understand by intuitively, just intuitively, that there is a stem cell here, and it can grow its clonal size over time. And it takes about 3 weeks just to have about half of the crypt filled by the progeny. And this is the case in the confetti system. And all the wild-type cells in our mutant behave just like the wild-type cells in the confetti system. Whereas the mutant cells that expresses Notch, KRAS, and PI3 kinase, they show a more rapid growth pattern. Up until here, I mean, this is interesting, but people already knew that oncogene can drive the clonal dynamics faster. So this was not really exciting to us. However, when Dr. Mingyu Yum has found this, we were very excited. So what he has found was the yellow clones in this mutant, they behave just like a wild-type cells normally, but when the yellow cells are positioned right next to the red cells, the clonal dynamics is somehow shifted toward the right side. And this means that somehow the dynamics of the yellow cells are changed because of the red cells. This means that the stem cell dynamics in the yellow is influenced by red, by some ligands and inhibitors that are secreted by the red mutant. So initially I was thinking simply that the clonal dynamics is faster in this tissue. We revisited the mathematical model. Excuse me. Okay, I will just continue. So the model tells us two important factors to consider for the time of the gain. So for this stem cell dynamics, we have to consider the number of stem cells and the speed of the proliferation. So we checked firstly the proliferation side, and different from my initial expectation, the proliferation rate of the yellow cells next to the red was not different from the yellow cells far away. However, the number of stem cells in the yellow cells next to the red showed clear downregulation. So normally we had 15 LGR5 positive cells, but in the yellow cells next to the red showed 20% reduction. And this 20% reduction is then multiplied again. So it's about 40 to 50% of reduction overall. The 2 times of time overall. So to understand what is the mechanism behind, and I have to go back once again, that the reduction of the stem cell, it means that somehow the red cells are suppressing the behavior of the yellow stem cells by secreting some inhibitory factors that are interfering the stem cell activity in the wild-type cells that are next to it. So we wanted to understand what's going on between the two. So we performed the single-cell RNA-seq. And just to cut the long story down into one slide, I have to just quickly tell you that we've done a very lengthy single-cell analysis, almost about 1 year, to figure out what's going on. So we analyzed all the epithelial cells, mesenchymal cells, and immune cells. And from the immune cells, in this very initial stage of the tumor development, we didn't see much differences. However, from the epithelial cells, we saw that when the KRAS or PI3 kinase is activated, from the Paneth cells and Tuft cells, we saw higher amount of BMP2 and BMP7. It means that red cells are secreting BMPs, which is a known factor that are interfering stem cell activity. And secondly, from the stromal cell type 2, we saw an interesting change that it loses the ISPO expression, and it secretes instead Wnt inhibitory factor. So it lose the Wnt antagonist, agonist, and it is secreting Wnt antagonist, which means that the stromal cells in the PI3 kinase model is somehow sabotaged, and it cannot support the Wnt activity anymore. So we wanted to check out whether this theory that we learned from the single-cell RNA-seq is real. So we checked whether KRAS is indeed secreting BMP7 as well as PI3 kinase model, and whether the stromal cells in the PI3 kinase model is indeed compromised. So we firstly checked in the organoid by taking out Nogin and R-spondin. So Nogin is basically an inhibitor against the BMP. So if you elevate BMP signal, the stem cell suffers. If you elevate BMP signal as well as inhibiting the Wnt signal, stem cell suffers more. However, when we have introduced these wild-type mesenchymal cells back into this culture, then we can kind of rescue the phenotype. However, the same mesenchymal cells from the PI3 kinase model cannot support the dish function anymore. So this was a very simple data we've got from organoid experiment to show that our theory is probably more or less correct. We've done also many, in situ data analysis, etc., to show that what we have observed from the single-cell RNA-seq is indeed real by histology. So there are many data in our paper. But I cannot show you everything, so I'm going to show you one very important in vivo analysis. So we also wanted to check whether BMP is indeed the most important factor for the crosstalk. So we basically performed the same lineage tracing while treating this LDN inhibitor that blocks the BMP receptor. And as you see here, the yellow clones that is next to the red actually showed faster monoclonal ligation compared to the conjective control, but with the LDN, this elevation was down, and it was same in the PI3 kinase model. The elevation was again down by LDN. So this was a very nice confirmation that the BMP signal is indeed the main factor that mediates the crosstalk. So this is the summary, and the paper is published last year in Nature. So if you like to know more about this, then please go to this paper and read all the details. The main conclusion we have is that these mutant cells, they are secreting BMP to suppress the stem cell activity in the nearby wild-type cells. And this is very striking because of the clonal dynamics. Even though we have a mutant cells here, there is a possibility that the mutant cell can be simply wiped out by losing the competition. But if they secrete BMPs to suppress wild-type cells around, that the chance for the mutant cell survive in this tissue competition is much higher. So we believe that this is the way how the oncogenic clone survives in the presence of millions and zillions of wild-type cells around. So I'm going to shift my gears to two other stories, and this is going to be much shorter. The first one is about conditional intron, and the reason that we started working on this is because conditional knockout is still very complicated, even after the introduction of CRISPR-Cas system. So people have already suggested to do double cuts by CRISPR and double insertion of LOXP sites in each side of the exon for conditional knockout in 2013. But then this was proven to be very inefficient by GRU, that it works just about less than 1% of the cases. So it means that this strategy cannot be used in daily life. So instead, we wanted to achieve the conditional knockout simply by cutting one site and inserting one segment. And for that purpose, we developed an intron, which is fully functional. So after the introduction of the SCON intron, you can still get protein expression. And interestingly, the SCON intron contains this LOXP site, which can then be recombined by pre-recombinase. And after the recombination, you lose this part, and this will then introduce a stop codon so that you can generate truncated protein instead. So we confirmed that it's working nicely in 293 cells first. And another very important point is that this EGFP, the native EGFP expression, is similar to the EGFP expression that contains SCON intron, which is now confirmed in the flow cytometry on the right side. The reason that I'm showing this is because if the SCON intron is not fully functional, there is a possibility to lose the GP expression a little bit, but then using this intron in mouse genetics is not so good idea. But our intron performed so well, so we started working with the mouse immediately. So here I'm showing you an example of a lethal gene, which is beta-catenin. Beta-catenin's gene name is CTNNB1. And for knocking out beta-catenin, we have utilized this SCON intron, which is just about 189 base pair. So the core part is 189 base pair, and we can put around 50 base pair of homology arms. And even after that, this is still within 300 base pair, which is fully synthesizable, which means that you can ask to a company to bring this template. So what the student can do is you can buy Cas9 protein or mRNA. You can also buy gRNA from a company, and you can also order this oligo template from a company. So because of all this commercial product, now the design of the conditional allele is completely digital. So once you know the sequence information, you can basically order them. So my student ordered all the reagents. After mixing them in water, he gave the reagent to our technician who does the microinjection into the mouse zygote. And after that, we got a pub. We got pubs and confirmed integration using sequencing. And thanks to our luck, we were able to get this homozygote, which was very exciting because this is a lethal gene, and even after this modification, if the homozygote can survive, which means that the allele is still fully functional, which is the beauty of conditional allele generation. So, so the mutants can survive with a good Mendelian ratio. And when we performed knockout in using conditional knockout method, we saw this data. So in the homozygote, the beta-catenin expression is very comparable to heterozygotes. However, after tamoxifen injection to activate CRE, we then lost all the beta-catenin expression in the entire epithelium. And as a result, we also have lost proliferating progenitors because beta-catenin is important for the Wnt pathway, which is important for the progenitor maintenance. So we can then wipe out all the epithelia. So this was a very nice confirmation that the conditional knockout strategy works with the SCON intron. So up until now, we have generated many conditional alleles based on this. So these are about 9 mice, and we currently have about 13 different lines using this method. And this has done by single PhD students on a site just in a year. And some of the genes are, I think 2/3 of the genes are lethal genes, but they survive as a homozygosity. So we are very excited that the Intron system seems to be working in a very robust way. And we have also tested this Intron in various species. For example, ratas, and this is pig, and 2 different species of monkey. And the conclusion is SCORN functions as it should. And there is another short conditional Intron, which is named as a decay. And this decay always underperforming than SCORN in various cell lines, which means that if you insert decay instead of SCORN, you get lower GFP expression because this decay Intron is not fully functional. So I would suggest you strongly that you have to use SCORN. And SCORN can also be utilized by this aqueous species. So you can also apply this to Xenopus and zebrafish. But what we have found is that, especially in zebrafish, the GFP signal is now dimming. And this is probably a bad sign because this means that at least in zebrafish, the SCORN Intron is not as functional. So we are currently thinking about developing another one for fish models. So to be able to apply SCORN, there are some rules, and we apply this to the entire genes in the mouse, rats, macaque, marmoset, monkeys, and medaca fish. And the conclusion is that almost from 80 to 90% of the genes can be targeted by SCORN Intron. And SCORN Intron can be also used for single exon genes, where the conventional conditional knockout strategy was not so easy to apply. You can also kill out only the splicing forms. And we can also introduce truncations in the last exons as we want to. So there are more nice applications we have with the SCORN strategy. So we are hoping currently that we set up new collaborations with the people working with the pig and marmoset or macaque monkeys in Korea and in other countries. So we hope that, in about short time, like in a few years, we can see primate models with a SCORN conditional Intron. So I'm going to show you with about 10 minutes about the Mosaic knockout system. So now some of the students might be wondering why Mosaic analysis and SCORN, because the two doesn't seem to be going together well. But if you have watched this movie, something strange can happen. So this is exactly what happened in the lab. So we have combined the two systems by modifying the system a little bit. So we firstly generated red to flip system, which means that instead of oncogenes, we now have a flip recombinase in the downstream of RP. So only the red clones will be expressing flip recombinase. And to use this flip recombinase, we also generated SCORN FRT system, where instead of LOXP, we have a FRT sequence for FLP-mediated genetic knockouts. And this works as good as a SCORN LOXP system. So the student generates this red to flip system, which took about 2 years. And then he also generated SOX2 SCORN system by introducing the SCORN Intron into SOX2 allele, which is a single exon gene. So this is the very nice example that SCORN works in single exon genes. And when it became a homozygote, they are still viable, which is a very good news because SOX2 is also known as a very important lethal genes. And we get homozygote in about with a Mendelian ratio, which means that they doesn't seem to have much problem. So we now have combined everything together in a mouse. So it has a Rosa CRE-ER2, which means that CRE-ER expression is in the entire body. We then have a red to FLP for FLP specific, the red specific expression of FLP recombinase. And then this recombinase can knock out SOX2 via the SCORN FRT system. So we performed 1 month lineage tracing and looked into the esophageal tissue. So we saw that the yellow cells, which is wild type, can nicely generate all this progeny within this 1 month of time. However, in the mutant, this is the red mutant, they have lost SOX2 expression, and they do not contribute to the lineage tracing, which means that after knocking out SOX2, they do not show the stem cell behavior anymore. So the wild-type clones, they generate many progenies when we analyzed in total 32 wild-type clones, but in the observed 23 mutant clones, we didn't see as such. So this was very clear. So at the moment, we are collecting more examples of mosaic knockouts by performing reporter gene expression. So only the red-green signals are coming from the red mutants. And this is the case of APC knockout in a red clone specific manner. So after losing the APC tumor suppressor, we see the elevation of beta-catenin only in the red clone specific manner. So I hope that we can generate more mosaic genetics knockout models using this system. And I'm very proud to say that this is the more or less the first very feasible mosaic knockout system and overexpress system in the mouse. So back in the days, we had a Madame system, but this system requires interchromosomal translocation. So don't even think about using it because such an inefficient system. And there was a dual FEG mosaic system introduced by Cell in 2017, but this is entirely only for overexpressions. So if you want to do a true conditional knockout, I think we only have our RED2 FLP system with just SCON FRT as a single option. So if this works as planned as we have, then we can perform sconing in the Cree homozygote and in the RED2 FLP homozygote colonies. And after the injection, we can find Cree homozygote SCON mouse and RED2 FLP homozygote SCON mouse. And by crossing the two, we can generate Cree containing FLP mouse with a SCON with a variable genotypes. This means that in about 6 months of time, so just 2 generations of a mouse, we can perform mosaic genetics in the mouse system. That would be, super fantastic. So the main conclusion we have here is that we've been envying fly geneticists that they have such a nice mosaic system in the fly, but I can now tell you that mouse can fly too. So as a summary, I have shown today that how we can perform mosaic genetics by using RED2 Onco and RED2 FLP system together with SCON FRT. The SCON system will help how we perform conditional knockout in primate models and other species. And because of the SCON, we now know the completely functional intron. And by generating a stopper within this intron, we can firstly generate an intron that is inactivated, but then we can remove the stopper to convert this inactive one into an active one. And by doing so, I think we can perform reversible genetics in about 2 years because we are already developing this. So with that, I'd like to thank for your attention and also my people, especially Sam, Christian, Jacek, and Peter. And this is the team in Vienna when we had a nice dinner together in my old flat. If you have any question, I'm happy to answer. Thank you very much.
Okay, thank you very much. So any questions? Either you can speak Korean or English or Chinese.
I don't understand Chinese, but if somebody can translate. But Korean and English are welcome. Chinese too.
Okay. Questions? So I'm not actually good at those the mouse analysis, but I think regarding the first topic, you nicely present the kind of image of each cell and then their image. So those are the conducted by just the microscopy, and methodology wise, how can you analyze those each cell in the organ?
Yeah, so what we do is if there is a tissue, so firstly we give a tamoxifen injection to the mouse to activate the system, and then we get differently colored clones in a target tissue, and after waiting some time, we then expect the clonal behavior changed, and we isolate the organ, and after fixation we cut them. Usually about 100 to 150 micrometer of a thickness is a thick tissue. This is actually necessary to preserve the behavior, the changes of their behavior, because sometimes the tissue can be mistily oriented in the histology. So 150 micrometer is a safe thickness. And this requires clearing. So we then perform tissue clearing using some method, and then we put them under the confocal microscope, or even sometimes multiphoton microscope, to be able to visualize this. So it is actually true that the imaging part of the work is also very tough, which can take about sometimes like 10 days in total.
Okay. So those part, you are mostly focusing on the intestine organ, right? Region, right?
Yeah, so compared to other ones, intestine is relatively easy for imaging and also tissue clearing, because the mouse intestine is kind of very thin. They are not like the pig intestine and cow intestine that we enjoy. They are very tough and thick, but mouse intestine is very simple. So it's sometimes just to look at the crypt, we don't even need to do the clearing.
Okay, I have a question. It's a really great talk. I have some concern about the stability issue about the labeled cell. When you introduce some labeling, when you introduce the GFP, RFP, whatever, so how long they stay there? So is there any concern about deletion of those kind of the labeled fragment?
So in the other study, we also used the confetti system, and I can tell you that the label is pretty stable, even up to 1 year. There is a possibility that this is a foreign protein, so there could be an immune reaction to it. But I think somehow the GP and the RAPs are well tolerated by the mouse. So if we analyze it very carefully, we will see some immune reaction, but it seems to be okay. So in my experience, there was not so much of difficulties. Yeah, so practically it's pretty stable.
Okay, thank you.
Okay, so one more question is, so have you ever had a chance to look at the other organs, like a neuron, a brain, or after mosaic expression?
Yeah, so I think it's a very good question, because since we are only mainly interested in intestine and the stomach,
Yes.
So we've been only using this to the two organs. I have a collaboration with some researchers to use it in the lung. And our lab has started working with the liver lately. But for the other organs, we don't have collaborators yet. One collaboration that is just about beginning is with the Professor Segyu Che in postdoc. He studies hair follicles and the skin epithelium.